Hello everyone. Now let us move to our next intestinal protozoal disease, that is Giardiasis. Introduction of Giardiasis. This disease is caused by the parasite Giardia lamblia. As you are aware, with the classification of the protozoa, that is amoeba, flagellates, ciliates and epicomplexa. Amongst these four, Giardia lamblia belongs to the class flagellates, specifically intestinal flagellates. This organism Giardia lamblia was named after Professor Giard of Paris and Dr. F. Lamblay of Prague. Giardia lamblia is worldwide in distribution and is the most common cause of both endemic as well as epidemic intestinal diseases in humans. Also, it is more commonly found in warm temperate climates of the tropics and subtropics. And the transmission of this organism is via fecal-oral route by contaminated water and food. Now let us have a look at the historical aspect of Giardia lamblia. Giardia lamblia was first discovered by Antony van Leeuwenhoek in the year 1681. Antony van Leeuwenhoek was observing his own diarrheal stools under the microscope and he discovered the trophozoid of Giardia in his own stools. And this is the way he described how the trophozoid of Giardia looks. He says, they are animalcules moving very prettily. Their bodies were somewhat longer than broad, and their belly, which was flat-like, furnished with sundry little paws. And albeit they made a quick motion with their paws, yet for all that they made but slow progress. We will see how the trophozoids are described later. Giardia lamblia inhabits in the crypts of small intestine and they occur in two forms. First is trophozoid and the other is cyst. The trophozoid is the pathogenic form and the cyst is the infective as well as the diagnostic form. Now let us have a look at both the forms. Starting with trophozoid. Trophozoid measures around 14 by 7 micrometer. As you can see here, this picture here, this is the front look of trophozoid and over here is the lateral view of the trophozoid. In the front view of the trophozoid, the shape of this trophozoid seems to be a pear shape or teardrop shape or a tennis racket shape, while in the lateral view it appears as a spoon or sickle shape. Let us see some characteristic features of these trophozoids. As you can see here, this trophozoid has got a pair of sucking ventral disc. As you can see here, this sucking disc helps the trophozoid to get attached to the intestinal mucosa. Also, it has got a pair of nuclei, as you can see, and a very special feature to the class which it belongs to, that is the flagellates. It has got 4 pairs of flagellates. As you can see here, pair 1, the second pair, the third pair and the fourth pair. So it has got 4 pairs of flagella. Then there is a pair of exonyms, which are the intracellular portion of flagella. And this flagella gives motility to the trophozoid, typically called as falling leaf motility. And coming to the cyst of Giardia, as you can see here, the cyst is oval shaped, measures around 8 to 12 micrometer It has got a thick cyst wall which is unstained, which remains unstained on iodine mount. It has got 4 nucleus, axoneme and some remains of the flagella. The life cycle of Giardia lamblia is simple, that is, it requires only one host, and the mode of transmission is ingestion of food that is contaminated with cyst. Once a person ingests food or water that is contaminated with the infective cyst, this cyst then undergoes excistation in the duodenum within 30 minutes of its entry. In excistation, 2 trophozoites are released from each cyst. These trophozoites then multiply by longitudinal binary fission. Then, with the help of the ventral sucking disc, these trophozoites gets adhered to the intestinal mucosa. Now, in the active stage of the disease, these trophozoites get directly excreted in the stool of the patient because of the sloughing of intestinal epithelial cells. Then gradually, when the trophozoites they pass down to the jejunum, encystation begins. That is, the cysts are formed, and then these cysts they get excreted in the feces and can survive in the environment and then are infective to the human being. The same thing is depicted over here. The cyst enters the small intestine, then multiplies by longitudinal binary fission, gets adhered with the help of the ventral disc and undergoes encystation. The cysts are then excreted in the feces to the environment, which becomes infective to man. Pathogenesis of giardiasis. As few as 10 to 25 cysts can initiate the infection of giardia. Most commonly, children are affected, though there are some other high risk groups also, as for example, immunodeficient individuals, individuals with poor hygiene, etc. As we earlier saw, the pathogenic form of giardia, that is the trophozoid, it gets attached to the epithelium of the small intestine and it causes disruption of these epithelial cells. This process might cause increased permeability and malabsorption. The malabsorption could be of fat, which leads to steatorrhea. It could be of disaccharides, which can cause lactose intolerance, or it could be malabsorption of vitamin A and B12. Giardia lamblia, it can undergo very frequent antigenic variation because it has got a surface protein that is called as variant surface protein, and this protein helps this parasite to evade the host immune system, because of which there could be recurrent and chronic illness. Clinical features of giardiasis can be divided in 3 stages: asymptomatic, acute giardiasis and chronic giardiasis. Most of the infected persons are asymptomatic. They merely harbor the cyst in the gut and spread the infection. While in acute giardiasis, there could be some general symptoms of fever, anemia, allergy, and there could be some intestinal symptoms of enteritis that can lead to fatty diarrhea, also called as steatorrhea. In steatorrhea, the diarrhea is often foul smelling with fat and mucus, but there is no blood. Giardia is also an important cause of traveler's diarrhea. And in chronic giardiasis, there are recurrent episodes of foul smelling diarrhea, sulfurous belching with rotten egg taste, and profound weight loss, which might lead to growth retardation. Also, giardiasis can affect the gallbladder, causing cholecystopathy, which might cause jaundice. Laboratory diagnosis of giardiasis. In lab diagnosis, stool microscopic observation remains the gold standard for the diagnosis of giardiasis. It detects both the forms, trophozoite and the cyst, the morphology of which we have already seen. For stool examination, normal saline and iodine mount can be used, as well as permanent staining is also used. Remember, in this case also, stool sample has to be repeated intermittently. When the stool examination is negative, one can also go for duodenal aspiration. There is a peculiar test for the diagnosis of giardia lamblia, that is called as entero test. In entero test, as shown in this picture, there is a gelatin capsule which is attached to a thread containing a weight. One end of this thread is attached to the patient's cheek, and then the capsule is swallowed. The capsule gets dissolved in the stomach, releasing the thread which is carried to the duodenum. It gets unfolded and it takes up the duodenal contents. Then after 4 hours, this thread is withdrawn and kept in saline. This process will release all the trophozoites from the duodenal contents, and this can then be detected microscopically by either wet mount or stained smear. Then there are antigen and antibody detection tests also available, ELISA and immunochromatographic tests. As you can see here in the picture, the cyst form of giardia lamblia and the trophozoite form of giardia lamblia, and these are the stained smears. This is the trophozoite form and the cyst form. Remember, the trophozoite form of giardia typically presents a type of motility that is called as falling leaf motility. Now let us see some opportunistic intestinal Coccidian parasitic infections. Now these are the intestinal parasites that belong to the class Apicomplexa. Some of the parasites under this class are Cryptosporidium, Cyclospora and Cystoisospora. All these parasites are opportunistic, that is, they affect severely in the immunocompromised host. In immunocompromised host, as in case of AIDS or HIV or patients on chemotherapy or some other immunodeficiency situation, these organisms can cause chronic persistent profuse diarrhea leading to severe weight loss. While in case of immunocompetent host, it causes only vague symptoms of diarrhea, nausea and fever, and it is mostly self-limiting. Also, they can cause 2 to 6% cases of traveler's diarrhea associated with traveling to developing countries. For laboratory diagnosis of these Coccidian intestinal parasites, stool examination stays the mainstay of diagnosis, wherein wet mount and acid fast staining helps. Also, there are other methods like histopathology, antigen detection in stool, antibody detection, and molecular methods. Here is the picture of modified Zn staining, that is the modified acid fast staining. The round shape acid fast are cryptosporidium, and these oval shape acid fast structure are isospora. Thank you.